Hello friends, welcome to my channel. I am Vidya Dhere. Today we are going to learn about angiotensin, bradykinin and substances P, that's peptides. Let's go. We will see one by one. First is angiotensin. Angiotensin is a peptide hormone that causes vasoconstriction and an increase in blood pressure. It is the part of the renin-angiotensin system which regulates blood pressure. We will see next slide. What is the renin-angiotensin system? This is the vasoconstriction, means constriction of blood vessels. Then, how the angiotensin produced? Kidney is the rich source of renin, where it is stored in granular juxtaglomerular cells in the walls of afferent arterioles in the form of prorenin. The prorenin in turn is obtained by intracellular proteolytic of an inactive precursor, preprorenin. Both prorenin and preprorenin are also present in plasma. Then angiotensin first is produced by the action of a renin on a problem called angiotensinogen, which is formed by the liver. Renin is the enzyme produced by the kidneys. Angiotensin first is transformed into angiotensin second, means angiotensin 2, in the blood by the action of an angiotensin converting enzyme, that is ACE. Angiotensin also stimulates the release of aldosterone from the adrenal cortex to promote sodium retention by kidneys. Then we have to see renin-angiotensin system. The renin-angiotensin system is an important part of the homeostatic mechanisms in the body. It works to maintain the blood pressure at the normal level. It also regulates the electrolytic balance by controlling aldosterone secretion by adrenal cortex. We have to see how the renin-angiotensin system works. Renin catalyzes the conversion of angiotensinogen into angiotensin first. Angiotensin first, which has less intrinsic activity, is converted to more active form angiotensinogen second by ACE, that is angiotensin converting enzyme. This enzyme also catabolizes bradykinin to inactivate fragments, and hence it is also known as kinase 2. Angiotensin 2 is one of the most potent vasoconstrictor agent. The components of a renin-angiotensin system get activated by lowering blood volume, low renal pressure, and low sodium ion concentration in the plasma. Then, pharmacology of angiotensin means effect of the angiotensin on our body. First, we will see effect on the central nervous system. Some amount of angiotensin stimulates the central sympathetic transmission. The feeling of thirst is potentiated. It also activates the central mechanism for the release of antidiuretic hormone. The resultant increase in the water intake and efferent sympathetic activity may lead to the hypertensive state. Angiotensin also exerts a direct stimulant action on a sympathetic ganglia and adrenal medulla. Then, next we will see effect of angiotensin on the peripheral nervous system. Angiotensin directly stimulates the peripheral sympathetic nervous system, resulting into vasoconstriction. You all know what is a vasoconstriction. Then, effect on the adrenal medulla. The increase in the rate of release of catecholamines in reported the rate of synthesis and release of aldosterone, resulting into increased retention of sodium and exertion of potassium and hydrogen ions. Then, effect on a cardiovascular system. Angiotensin also exerts a positive inotropic and chronotropic effect on the cardiac muscles. Angiotensin causes an increase in vascular permeability in large arteries, resulting into edema formation. Then, effect on the extravascular smooth muscles. The diversified effects ranging from contraction to relaxation on various smooth muscles are exerted by angiotensin. Then, effect on kidney and urine formation. In normal person, angiotensin has an antidiuretic action due to the decrease in the rate of glomerular filtration. Then next, mechanism of action. The presence of a specific receptor sites for angiotensin in certain tissue, that is chromatin cells in adrenal medulla, has been suspected. There is an evidence that the action of angiotensin on heart and smooth muscles may involve calcium-dependent mechanism. In some cases, the activation of a cyclic nucleotide is responsible for angiotensin-induced responses. The next, drugs acting on angiotensin. First is captopril, enalapril, fosinopril, ramipril, lisinopril, perindopril, trandolapril, imidacloprid, and benazepril. I will describe action of a captopril. Inhibits the action of angiotensin 1 but not of angiotensin 2 and does not block angiotensin 2 receptors. Captopril-induced hypotension is a result of a decrease in total peripheral resistance. The arterioles dilate and compliance of larger arteries increases. Both systolic and diastolic blood pressure decreases. No effect on cardiac output. Captopril also inhibits the inactivation of bradykinin by inhibiting the action of kinase 2. As a result, bradykinin promotes vasodilation by decreasing peripheral vascular resistance. These all are the examples of drugs acting on angiotensin. These are the angiotensin antagonists: losartan, telmisartan, candesartan, valsartan, and irbesartan. Losartan is a competitive antagonist and inverse agonist of angiotensin 2. It is 10,000 times more active against angiotensin first and angiotensin second. It blocks overt action of angiotensin second, that is vasoconstriction, central and peripheral sympathetic stimulation, aldosterone release from adrenals, central action like thirst, vasopressin release. These all are the examples of angiotensin antagonists which inhibits the action of angiotensin. Then bradykinin. Bradykinin is an inflammatory mediator. It is a peptide that causes blood vessels to dilate via the release of a prostacyclin, nitric oxide, and endothelium-derived hyperpolarizing factors. Bradykinin is a physiologically and pharmacologically active peptide of the kinin group of proteins consisting of 9 amino acids. A class of drugs called angiotensin converting enzyme, that is ACE, inhibits the increased bradykinin levels by inhibiting its degradation, thereby increasing its blood pressure lowering effect. Angiotensin converting enzyme inhibitors are FDA approved for the treatment of hypertension and heart failure. Bradykinin is an inflammatory mediator. It is a peptide which causes blood vessel to dilate. Then next, how the bradykinin produced. The bradykinin receptor family is a group of G protein coupled receptors whose principal ligand is the protein bradykinin. There are two bradykinin receptors, the B1 receptor and the B2 receptor. Bradykinin is a major functional vasodilator produced by the kallikrein-kinin system. Bradykinin exerts its effect via B1 and B2 receptors. The bradykinin synthesizing enzyme, kinase 2, is identical to angiotensin converting enzyme which produces angiotensin 2. Then function of the bradykinin. Effect. Bradykinin is a potent endothelium-dependent vasodilator and mild diuretic which may cause a lowering of blood pressure. Then receptors. The B1 receptor, also called bradykinin receptor B1, is expressed only as a result of a tissue injury and is presumed to play a role in chronic pain. This receptor has been also described to play a role in inflammation. Next, role of a bradykinin. Angiotensin converting enzyme, also referred to as a kinase 2, is the principal enzyme responsible for the metabolism of bradykinin, a potent endothelium-dependent vasodilator. Bradykinin is a potent endothelium-dependent vasodilator. Bradykinin induces vasodilation by stimulating production of nitric acid, the arachidonic acid metabolism, prostacyclin, and PGE2, and endothelium-derived hyperpolarizing factors. Then, clinical use of the bradykinin. Bradykinin B2 receptor antagonist indicated for acute attacks of a hereditary angioedema. Shortly, we say that HAE. Recently, numerous BK receptor antagonists have been synthesized with prime aim to treat diseases caused by excessive kinin production. These diseases are rheumatoid arthritis, inflammatory diseases of a bowel, asthma, rhinitis, and sore throat, allergic reactions, pain, inflammatory skin disorders, endotoxic and anaphylactic shock, and coronary heart diseases. Then, next point, substance P means peptides. It is an undecapeptide first detected in the extracts of brain and gut by scientists Euler and Gadam in 1931. It is localized within nerves in the CNS and periphery, and hence also termed as a neuropeptides. In a CNS, it is present along with serotonin in certain neurons. Substance P stands for preparation of a neurokinin type 1 receptor, is distributed over cytoplasmic and nucleic membranes of many types of cells and many tissues and organs. It excites more cellular process. Substance P is synthesized by neurons and transported to synaptic vesicles. These are the neurons. How the peptides are synthesized. Peptides are synthesized through translation and transcription process. Substance P is synthesized in the endoplasmic reticulum, then transferred to the Golgi apparatus for the packaging, and finally transported to the cell membrane for exocyclic release. It is present in a high concentration in nerve endings in selected region of the mammalian brain. Substance P is synthesized by neurons and transported to synaptic vesicles. These are released by the depolarizing action of a calcium-dependent mechanism. When neurokinin 1 receptor is stimulated, then they can generate various second messengers, which triggers the various effects mechanisms that regulate cellular excitability and functions. Then next, functions of the peptides. First, these are all the functions of peptides. First is vasodilation. Substance P is a potent vasodilator. It is dependent on a nitric oxide release. It also has a bronchoconstrictive properties administered through the non-adrenergic, non-cholinergic nervous system. Then, inflammation. Substance P and other sensory neuropeptides can be released from the peripheral terminals of a sensory nerve fibers in the skin, muscle and joints. This release is involved in neurogenic inflammation, which is the local inflammatory response to certain types of infection or injury. Then, it causes pain. It is due to the transmission of a pain information into the central nervous system. Substance P coexists with the excitatory neurotransmitter glutamate in primary afferents that respond to pain stimulation. Then, It causes mood, anxiety, learning. Substance P is associated with the regulation of mood disorders, anxiety, stress, reinforcement, neurogenesis, respiratory rhythm, neurotoxicity, pain, and nociceptions. Then vomiting. You all know what is vomiting. It causes vomiting. Then cell growth, proliferation, angiogenesis, migration. It causes cell growth, proliferation, angiogenesis, and migration. Substance P stimulates cell growth in normal and cancer cells. Line cultures. Substance P promotes wound healing of non-healing ulcers in humans. Then we have seen these 3 points in today's lecture. Thank you for cooperating. Thank you.